R.C. Lai, the current president of Bay Area Taiwanese Biotech Association. Today, it's my great honor to have our keynote speaker, Dr. Tim Lu, join us for the biotech sections. So let me briefly introduce Dr. Tim Lu. So Dr. Lu earned his MD from Harvard University and his PhD in electrical and biomedical engineering from MIT, and it's a joint program between MIT and Harvard program. And since 2010, Dr. Lu has been an MIT faculty member in the Department of Biological Engineering and Electrical Engineering and Computer Science. In addition, Dr. Lu has been a co-founder and a scientific advisory board member to a number of biotech, including the one he's going to present today, the Senti Bio. With his extensive experience and great work in synthetic biology, he already received numerous awards, including NIH New Innovator Award and U.S. President Early Career Award for Scientists and Engineers. Without further ado, please join me to welcome Dr. Tim Lu.
Hello, can you hear me? Great. Thanks a lot for the opportunity to be here. It's really exciting to spend some time. This is, by the way, a great conference. I heard it was the first time you guys are doing such an integrated group meeting, so I think it's really exciting to be part of it. I was here for some of the previous sessions, and actually, as part of the introduction to what I'm going to talk about today, it's amazing to see, sort of, especially today in the heart of Silicon Valley, all the advances that have been made in software, hardware, leading to the ability to do crazy things like shoot rockets into the sky, all sorts of new fancy ways of processing data. A lot of that is driven by something really fundamental, which is that we have exponentially greater computing power available to us so that we can crunch data, we can analyze patterns and understand that. And clearly we're using that to do, things that make us happy, like, watch TikTok or shoot things to the sky. But actually, I wanted to tell you today that something very similar is happening in the drug discovery space. And that really is going to, we believe, make a huge impact in the future of human society and the way we treat diseases. So what's happening is essentially we are gaining a much greater and deeper understanding as to the genetic basis of disease. And that's really coming from the fact that we can read DNA and write DNA much faster than we used to. So I started in this field about 20 years ago. This is when the first Human Genome Project was announced. A lot of investment went into that at the time to just read the DNA that's inside of our cells. But since then, that has led to really, in many cases, exponential improvements as well. We can read DNA and write DNA much faster than we used to be able to do. And that's allowed us to start gathering much, much more deeper insights into what causes disease and also how we can treat disease. And so what I hope to share with you today as a just very simple story is one of the outcomes of that is that it's going to allow us to start building hopefully better medicines. I think we're all familiar with certain types of drugs that we take as we're growing up. You think about aspirin or Tylenol, very simple chemical drugs that essentially were discovered through digging around the dirt or isolating a plant and seeing what it does, to slightly more advanced drugs, the drugs that companies like Genentech make, biologic drugs, protein drugs that can treat diseases in much more targeted ways. And now we have a whole new wave of medicines that are coming, right? We have cell therapies. I'm going to talk about that today, where we can take living cells out of a person. We can genetically modify those cells and then put them back into the patient and use that to hunt down disease. We have gene therapies, where you can take a virus and you can program that virus so that it goes into a patient and changes fundamentally, permanently their DNA to treat a disease in a sort of a permanent fashion. We have new vaccines. Obviously, we saw that through COVID. Within a few days of the sequence coming out, you could turn around and actually make a new sequence and make a new RNA vaccine. So these sorts of breakthroughs are, I think, just the tip of the iceberg, but really are enabled by our increasing ability to read and write DNA. That's at the heart of what we're trying to do. So I wanted to share with you today a bit of the work that we're doing at Senti. We are just one example of what's happening in this field. There's many other efforts going on. But if you can jump to slide 5, maybe I'll start there. So Senti, so I moved to the Bay Area, several years ago from Boston to start this company, Senti Bio. And really the idea was to really take advantage of some of those advancements we talked about earlier. So we're actually based out in South San Francisco, not too far from here. And what we wanted to do was actually bring the power of reading DNA and writing DNA to make smarter medicines. Right? We want medicines that are more accurate, that are more controllable, and ultimately medicines that are more effective. And so we started to look at what are the sort of like core problems in terms of treating disease. Right? So I use cancer here as an example, just because I think most people are familiar with that as a big health challenge. One of the big challenges in cancer is how do you tell the difference between a cancer cell and a healthy tissue, right? We all probably know people who've taken chemotherapy. Chemotherapy is very toxic. Your hair falls out. You have, GI symptoms, etc. And yes, you kill the tumor cell, but you also kill a lot of your healthy cells. And that's the challenge we talk about, up here, which is, you get in the treatment of cancer a lot of on-target but off-tumor toxicity. What that means is, you're trying to hit something, but it's actually not found on the tumor cell, it's found on some healthy tissue, and you're going to kill that healthy tissue. That's a big issue. Number 2 is the existing cancer drugs we have may be effective for a period of time, but cancer is really smart. Like cancer finds ways of evolving and getting around the treatment, and that leads to things like tumor relapse. There are some new drugs that have been developed to try to treat cancer using the immune system. But as a result of trying to activate your own body's immune system to attack cancer cells, you can generate systemic toxicity as well. And those are side effects that patients don't want. And lastly, I think I sort of talked about this imprecise tropism. Again, this targeting idea is a very difficult thing for oncology. A lot of this comes from the fact that our existing drugs are just not very smart. So the way I like to think about existing drugs is they're sort of like a railroad, right? You can have a car, you can have a train on a railroad. It goes in one direction and one direction only. It may go really fast in that direction. It may be great in going that direction, but you can't really change what it does after it's programmed to do what it does. I think what we want to try to create for the future are drugs that are more like self-driving cars. Drugs that can go in the body, can detect what's going on, can go to the right place, can be turned on or off, and can make a decision as to, do I kill this cell or do I spare this cell? And so to do that, we need to actually be able to program greater intelligence into these medicines. Just like we heard about programming a chip or a rocket, we can do that in living cells today by changing the DNA and inserting new programs. We call these programs gene circuits. They're essentially made up of genes that we put together in such a way that they can do things like logic. It allows us to dial things up and down. For example, we can turn up how strong the drug is, or we can turn down how strong the drug is. We have gene circuits that act as sensors. They can detect, am I in the right tissue, or am I detecting the right signal? Or maybe I want to detect 2 signals or 3 signals at the same time before deciding what to do. And so that's really at the core of what we're trying to develop here at Senti. Next slide. To do this really requires an engineering approach. So the other thing that I think is quite interesting that's evolving in our field is that drug discovery is starting to move from essentially stochastic search process. In the past, we would screen billions of compounds, and hopefully we find one that did the thing we want. We don't necessarily know why it does the thing we want, but it did it, and so we took that forward and made drugs like aspirin out of it. I think what we're moving towards today is more of an engineering mindset. How do we actually forward design a medicine that actually has a certain function, has a certain design profile, a criteria, et cetera? And so to try to enable that, companies like ourselves, as well as others in the field, are trying to really leverage this engineering mindset. I think that's probably familiar to a lot of people here that we're doing in other fields, in mechanical and electrical engineering. But can we design, right? Leverage computational tools, leverage machine learning to actually study the data that we have today, come up with new designs that we think could be effective. Number 2 is build. We need to be able to build these in high throughput. So that's not just something that Senti is doing, but working together with other groups in the industry. I think my moderator later is coming from Twist. They build large libraries of things so that we can actually test them in high throughput. So we want to be able to characterize a lot of data. We don't want to just test one thing at a time, but can we test billions of things at a time and collect quantitative information about how this design is working, how this is not working. And then the most important part is learning from this. The learning aspect is not just man-made, but it's also computational, building algorithms that can digest the data and come up with new designs automatically. So this is the process we use to try to create our genetic circuits. There's a bunch of biological details here, but I think what I would say in general is we have 4 classes of circuits I talked about earlier. I'm going to show you some examples of how they work. The great thing about these circuits is that we can actually plug and play them into different cells or different gene therapy vehicles. So this is not just limited to a T cell in your body, but we can also do this for other cell types, including stem cells, natural killer cells, etc. So this is really this idea that this is software inside of different cellular modalities of the future. So if you go to the next slide, this is a standard pipeline slide you might see if you look at any biotech companies. At the end of the day, our goal is to move these things into the clinic and to treat patients. We are targeting to file what we call IND next year for 2 of our programs. For those of you who are not in the field, an IND basically means you submit an application to the FDA, and if they basically allow you to go forward within 30 days, you basically know about that and you can move forward into human clinical studies. So we have a set of programs starting from Senti 202 all the way down to Senti 311, so the first half of this slide, all focused on cancer applications. And one of the reasons why we've chosen internally to focus on cancer is it's obviously a huge unmet need. One of the benefits of treating cancer patients is that you can actually go directly and try the drug in those patients directly. In other disease areas, you might have to test the drug in some healthy person first before you can actually treat the patient. So it allows us to actually hopefully speed up our cycle times in terms of doing this sort of testing. The other thing is below the sort of middle line here, we also have a couple collaborations with other large pharma companies. Spark is a subsidiary of Roche Pharmaceuticals. Blue Rock is a subsidiary of Bayer. I won't go into details because of time today, but just to say that we are developing this sort of gene circuit software for other companies as well. Companies that are developing gene therapies for rare disease or who are developing regenerative medicine applications. As a relatively small company ourselves, we aren't able to do those clinical studies all on our own. And so we want to work with some of the sort of the best and brightest out there to try to get this technology into clinical studies as soon as possible. Okay, so let's jump forward 2 slides, please. All right, so let me tell you a bit of details. Again, I'm going to try to give you, for those who are sort of not well steeped in this field, give you a bit of an overview of why we're so excited about where this field is going. We work on a specific type of cell called an NK cell. So an NK cell stands for a natural killer cell. Natural killer cells are a specific type of immune cell that can be found in everyone's body here. The job of a natural killer cell normally is to detect abnormal things that are happening. So for example, if you get infected by a virus, an NK cell typically will recognize that that cell got infected and will kill it. NK cells also provide one of the first lines of surveillance for cancer. So, we are always developing little pieces of cancer in our body all the time. Usually our immune system can recognize that something has gone wrong, get rid of it, and natural killer cells are part of that. One of the exciting things about natural killer cells, especially in the last few years, is that they've been used as a new type of medicine. So what you can do is you can take natural killer cells from a healthy person. I can just go donate blood. Someone can isolate my natural killer cells, and they can expand them. So they can make, from a small vial of my blood, many, many, many vials of NK cells that can be used to treat a patient. So number one, that allows us to create potentially a large supply of these cells for downstream drugs. Number two is that when people have now engineered NK cells, so you can basically change the genes inside of an NK cell, and there's a couple companies as well as MD Anderson have done recent clinical studies. When they put these NK cells back in the body, these cells tend to be pretty safe. You can use them potentially in an outpatient setting. And they also seem to have good effectiveness. They have this ability to kill tumor cells sort of endogenously. So if you look on the right-hand side here, some of the benefits of these off-the-shelf NK cells is good safety, your ability to manufacture them. But there's still some challenges that are sort of left. One is, how do we make the cells better, more powerful, right? We want these NK cells to expand and persist in the body and really be as good as possible as sort of like killers of tumors. Number two is, how do we get them to recognize more intelligently different tumor targets? And also I'll show you some examples of where this sort of synthetic biology can really try to make these NK cells better. So let's go for the next slide, please. All right. This is a little bit detailed, but maybe let's skip over. Let's go to the next slide, 11. So one of the things that we do with our NK cells is we actually put them into tests to see how well they can actually kill tumors. Right, so this is actually a picture on the right-hand side. Those 3 pictures are microscope images. The red cells are tumor cells, essentially. And so what we do is we actually monitor, over 196 hours, how many tumor cells are left. And so what we can do is actually genetically modify these NK cells with several different things. So we can put in what's known as a CAR. A CAR is basically a protein inside of the cell that tells the NK cell what they should be killing. Right? So that number 1 is it's a homing beacon for this is a tumor cell. Number 2 is we can engineer them with something called IL-15, CR-IL-15. CR-IL-15 is a protein that actually stimulates NK cells and sort of makes them really happy and gets them to be effective killers. We have designed our own version of IL-15, which makes it, we think, very, very potent as a way of stimulating the NK cells. And we've tested them in experiments. Perhaps I'll just direct you to the experiment on the right-hand side here. So the way this experiment works is over 196 hours, we actually inject fresh cancer cells into the population. Basically, what we're trying to test here is how good are the engineered cells at killing tumor cells. This is called a serial killing assay, and I'll show you why that's the case. So if you look at this graph, diagram here. So if you look at this graph here, in yellow, these are tumor cells alone. They just keep growing, right? In the black, in the green, and the pink are basically NK cells that are delivered into the culture. So you can see in this first round of killing between hour 0 and 96, basically all of these NK cells are good at killing the tumor cells. However, by the second round of killing, when we add fresh NK cells at hour 96, you can see that 2 out of 3 of those NK cells actually sort of lose their potency. They get tired, right? They're not set up to do the hard work of continuing to kill tumor cells. So one of those is the unengineered NK cells. So the natural NK cells sort of peter out over time. Also in the pink, which is a sort of a wild type control. But the senti NK cells in blue, basically due to the engineering we've done, sort of repeatedly are able to kill by that second round. And that persistence continues to the third round. So by basically introducing 2 genes into the cell, we were able to actually significantly boost the activity of these NK cells and their ability to kill cancer. So this is one example of the sort of genetic engineering that we can do with these products. Next slide. So let me give you now an example of how we turn this into a real product. Okay, this is probably the main example I'll focus on today. So Senti 202 is focused on a disease called acute myeloid leukemia, or AML. It's a blood cancer that unfortunately does not have a lot of great treatment options. So go to the next slide, please, and I'll show you how this works. So AML is a disease that is not well treated. Basically, patients with relapsed refractory AML probably have less than a year, half a year to survive, unfortunately. There's basically one major problem, or one significant problem in AML that people have not been able to solve, and we call that target heterogeneity. So again, I mentioned earlier, one of the key challenges in cancer is how do you tell the difference between a cancer cell and a healthy cell? It's actually not easy to do that, And the reason for that is cancer cells sort of oftentimes share signatures with healthy cells. They sort of look the same. And so as a result, it's been hard to find a very clean target for a lot of cancers, including AML. None of the current targets that people are going after are expressed on all of the AML cells. So think about it. If you kill 50% of the cancer cells, the other 50% will just keep growing. So that's a problem. The second problem is many of the targets in AML that people want to go after are also expressed on healthy cells. So that means as a doctor, when you start treating a patient, you might kill off, 50% of those cancer cells, but you make the patient so sick that they don't want to continue with the therapy. And so that's another significant issue. This all results from the idea that there's not a clean target. And so one of the simple ideas we've had is, let's take a concept from electrical engineering. Why not design a product that can detect 2 things or 3 things and make a logic gate out of it, make a simple decision whether I should kill or not. That actually is pretty hard to do. But we've created essentially a logic gate that does this, that has an OR function and a NOT function. I'll show you in the next slide. So this is a picture of a cell. So this is our cartoon of what's going on. In reality, obviously, it's much more complex. But on the left-hand side, this is the engineered cell. Okay, so in blue, this is an NK cell. The NK cell has 2 major things that I want to point you to. It's got these 2 receptors, so the top receptor and the bottom receptor. Okay. The way I like to think about this is this top receptor is the gas pedal, and the bottom receptor is the brake. Right? So the top receptor is responsible for killing cancer cells. And so you can see here this top receptor actually binds to 2 different things. It has like 2 different recognition elements. And it basically recognizes 2 different targets on the cancer cell. Okay, so number one, that's what we call the OR gate, very simple way of programming an NK cell to target cancer based on whether it has one of two antigens. Either one of those antigens will allow it to be killed. And that's our way to try to reduce the likelihood of tumor relapse, because imagine having a complex cancer, maybe 50% of the cells have target A, maybe 50% of the cells have target B. If you only went after one of them, you're going to miss a lot. But if you go after two, simple concept, you'll probably hit more of them. The right-hand side shows how the break works. So the second receptor, which is this thing in purple, recognizes what we call a protective antigen. And so a protective antigen is a protein. It's sort of a signature that's expressed on the healthy cell, but not expressed on the cancer cell. So it's sort of like the opposite of what we're looking for. And so if this NK cell, when it's floating around through the body, happens to encounter this protective antigen, it says, okay, I know this is a healthy cell. Even if it sort of looks like a tumor cell, I'm going to leave it alone. And so the way it works is this purple receptor is engineered to block the activating receptor. So it basically acts as a brake on the system. So on a cartoon, this looks really simple, but in reality, it takes a long time. It took us quite a while to actually optimize this technology. The original concept of this was sort of created by several of our scientific founders at MIT and BU, basically where they showed that they had, basically identified certain proteins that could either turn on or turn off the NK cell. And so what we did at Senti was take that concept and really industrialize it. We went through our design, build, test, learn cycle. We built hundreds of different variants of these proteins, characterized their function in a quantitative way, and that allowed us to repeatedly optimize and design to get to a candidate that we think has a lot of promise. So next slide. So we do a bunch of experiments in the biotech field to try to characterize the performance here. And this is oftentimes actually where the bottleneck, I think, ultimately is going to end up in our field. The ability for us to engineer and design large data sets is really accelerating. At the end of the day, though, we are still getting bottlenecked in our pathway to the clinic, right? So that's the next problem to solve in the future, and we can talk about that perhaps later. But assays like this, these are 5 different AML samples. They're sort of coded differently, and they have these different M subtypes to them. There's just a way of just describing where they come from. But this is basically trying to show a very heterogeneous set of tumors. And we can show that our CAR NK cells, which are shown in the green here, do have significant increased killing compared to the native NK cells on their own. Next slide. We can also show significant extension of lifespan in animal models, right? And this is obviously what we hope to be able to see in humans, and part of the experimentation that we need to do to be able to justify why we're ready to take this into the clinic. Just to show you here, on the top 3 rows here, or all 3 of these rows, these are pictures of mice. Anywhere you see them lighting up with different shades of colors, that's basically a signal of cancer that's growing. So less blue is better, less color is better. And so what you can see is with the Senti or CAR NK cells, we can significantly increase the number of mice that are responders and basically extend their lifespan and reduce the tumor burden there. Next slide. All right, so that was showing our ability to kill tumor cells, those 2 experiments, right? We can kill tumor cells. We can do this both inside of animal models. We can do them across different human samples. Now I want to show you that the brake works. Okay, so this is a little bit of a complex experiment, so I'm going to show you briefly just as an example of what I'm doing here. So in the middle, we have mice. So this is a mouse experiment. What we do is we inject 3 different populations of cells. Number 1, we inject our NK cells in blue. Number 2, we inject healthy cells. And number 3, we inject cancer cells, right? So if this gene circuit is actually working, what we wanted to do is actually tell the difference between a healthy cell and a cancer cell, right? So the way we do this is we can then take the blood out of these mice and we can characterize them and measure how many healthy cells are there, how many cancer cells are there. So the middle graph here is showing how this works. So we have 2 different populations here. We have our control cells, which don't have the NOT gate functioning, and we have our NOT gate cells. And then in green, we have our healthy cells, and in gray, we have our cancer cells. So hopefully what you can see here is with the NOT gate, you actually see a pretty big separation. We can basically keep the healthy cells alive, but we can kill the cancer cells. Whereas with the controls, basically they're sort of indiscriminately killing everything. On the right-hand side is just a plot of this. This basically shows that we can enrich for the number of healthy cells in the downstream population. All right, so perhaps the last slide I'll show here is next slide. So we've now shown that not only works in a mouse model, but we can also use this and actually try to protect human cells with this as well. Just as a detail, in AML, it's a blood cancer. And so one of the big challenges in treating blood cancers is you want to preserve the natural bone marrow, right? Your bone marrow produces all of the blood cells in your body. It's pretty important. A lot of the treatments, almost all the treatments we have today to treat, diseases like AML may have some effect on those normal blood cells, including a specific cell type called the HSC. So HSC means a hematopoietic stem cell. So this is the key cell type that we're trying to protect. So the data we've shown here is, again, repeating that similar experiment as I showed you earlier, but now with human cells. On the left-hand side here is showing that we can continue to kill AML cancer cells quite effectively with the NOT gate. But on the right-hand side, hopefully you can see that there is a reduction of our killing in the population of hematopoietic stem cells isolated from humans. Now you'll see that in this case, we've sort of calibrated this gene circuit to be about 50% protective. We think for biological reasons, that's a really good target. But we have basically design NOT gates that are stronger or weaker for any downstream application of interest. So this is an example of, I think, the types of engineering that we're really excited to try to implement into new medicines of the future. Hopefully what I wanted to share with you today is not necessarily the details of our products and sort of all the intricacies of what's going on there, but perhaps the broader picture, which is that the development of biotechnology has really accelerated over the last several decades. We started off with a large amount of our drugs being chemical drugs, and those are the ones that are frankly very cheap. We buy at the pharmacy. We use them to treat common illnesses. The next stage of those drugs became protein drugs, where we started to develop antibodies or other enzymes that could actually target specific biological pathways as our understanding of those diseases became clear. Today, we're in an era where we're starting to see an emergence of additional types of medicines, cells that we can use in the body, new types of vaccines, gene therapies, microbes. It's really been an explosion of new ways of trying to treat disease. And really at the heart of all of these new strategies is our ability to program what these things are doing. So I think why I'm excited to be sort of here, I think it feels like the early days actually of the semiconductor industry where we have this technology. The sort of logic gates we're building here are super simple, right? Like it's like a 2-input logic gate, 3-input logic gate. But we're able, I think, if this is successful, to tackle diseases in fundamentally new ways. And I think the opportunity for folks who are here, who are either in the biotech space or not, is that it's going to require really a concerted engineering effort to do this. There's opportunities for greater computational and data science people coming into this field to help us crank through all of this data that we're generating. There's opportunities for more traditional biologists to come in and really use those tools in much more powerful ways. And I think there's opportunities for investors or the folks who are trying to support this field going forward to really try to take these innovations and hopefully use these to try to treat diseases, important diseases that we all will suffer from one day in fundamentally new ways. So with that, thank you very much for your time. Looking forward to the panel discussion and chatting more in the future. Thanks a lot.